Here you can see the slides. So previously, in case of the anatomy, you've heard about the anatomical structure of the genital tracts, of both genital tracts, and then now we will see how they develop during the intrauterine life. In case of the next two lectures, we will have these topics. So first, I will give you a short introduction, and then we will talk about the regulation of the development. Then we will see three different levels of the developments. First, we will discuss the development of the gonads. Then we will discuss the development of the genital canals. And then after that, we will do the development of the external genitalia. Then at the end, we will talk about malformations causing disorders of the sexual development. Could you please turn off your microphone, please? Thank you. Then first we will start with this short introduction. The gender could be, so we could have many different types of gender. In this case, we focus only on these three genders. The others, of course, we will have more other types. I have written only this psychosexual, but we have more other genders, but this is not our work to clarify these genders. We focus on these three types of the genders: chromosomal and the genotypic or genetical gender means which which sex chromosomes are found in the cells of the individual. Genotypic, it's a little bit more precise because it does not cover only the chromosomes, but it focuses on genes, on specific genes related to the development of the reproductive system. The gonadal gender or gonadal sex means only the thing whether there's a testis or an ovary inside the body. Phenotypic indicates the external. Phenotypic gender is defined by the external appearance. If we see the first point, of course, the first point is always the fertilization. In case of the fertilization, the chromosomal gender is determined, that is defined. We have an egg cell here that contains always an X chromosome, like here. And then the sperm cell, the spermatozoon, will define the chromosomal gender of the zygote. And if the sperm cell, the spermatozoon, contains a Y chromosome, the zygote will have XY sex chromosomes. If the sperm cell or spermatozoon contains X chromosome, the zygote will contain XX sex chromosomes. At each level of the development, I mean the development of the gonads, the development of the genital tracts and the male and the external genitalia, we could always define an indifferent stage. And from indifferent stage, based on the factors which are related either to XY or either to XX zygotes and embryos, we could get either the male genital apparatus or the female genital apparatus. We will see the regulation. For defining this, we need one gene, and this gene is the key factor. This is called the SRY gene. The SRY gene is found on the short arm of the Y, and that is the gene here. And of course, it codes a protein like every other gene, and this protein could be named the SRY protein or the TDF. TDF means the testis determining factor. The testis determining factor, or SRY protein, is a transcription factor. This transcription factor will be expressed in the somatic support cells of the developing gonad, and these somatic support cells expressing the SRY will be later the Sertoli cells, and they are the precursor cells. They are the precursors of the Sertoli cells, and then if these somatic support cells express the SRY, they will differentiate into Sertoli cells. If the somatic support cells does not express SRY and will not have the TDF, they could not be Sertoli cells. They will become the ovarian follicular cells. So this is actually, therefore, I've said that this is the key factor. The Sertoli cells, developing from these precursor cells, from these somatic support cells, will start to produce the AMH. AMH is called the anti-Mullerian hormone. And anti-Mullerian hormone plays a very important role during the development. It will cause the regression of the Mullerian duct. And this is what we will see. Mullerian duct will give rise to the components of the female genital system. Hence, it is very important that in case of the male development, these structures should disappear. If we have the SRY, the male development will start, and this is a transcription factor forming a complex with another one. This is called the SF1, or steroidogenesis factor 1, and this complex will cause upregulation of transcription factors related to the testis development. For example, SOX9, but we will see the little bit the cascade, which is started by the SRY protein. So here, this sketch shows us also that if we have SRY gene, male development starts. If we don't have, female development will start. Schematically, you can see this here as well. So optimally, if we have an XY zygote, or XY sex chromosomes are found inside the embryo. On the Y chromosome, we have the SRY gene, and then testis determining factor will be produced. This testis determining factor, or SRY protein, we could say, will cause the conversion of the different gonad into testis. And then hence we will get the testis development. Inside the testis, testosterone and anti-Mullerian hormone. These two hormones will be produced, and they will define the male development. So we could say that this is an active development. For getting the testis development, the SRY gene is necessary. In case of the other version, if the embryo has XX sex chromosomes, where we do not have Y chromosome and we do not have SRY gene, the indifferent gonad will become the ovary. So we could say, as others have previously done it, then this is the passive way. But this is not passive, because the development of the female genital tract is also a very complicated process. But of course, it does not need the active contribution of the SRY gene and the SRY protein, or testis determining factor. If we see the molecular mechanism, in this case, we see on this side the male cascade, and here the female cascade. In case of the male cascade, of course, SRY is necessary. The SRY protein is encoded by the SRY gene. The SRY protein complexes with this SF1. SF1 is also a transcription factor. SF1 has also effect on its own on the transcription of this SOX9. SOX9 factor is very important from point of view of the male development. The SRY and the SF1 together with this complex formation have a very significant effect on the SOX9 upregulation. SOX9 upregulation is necessary for the male development. If we don't have any upregulated SOX9 factor, female development will occur. If we have SOX9 upregulation, male development will occur. SOX9 acts on different genes related to the male development. Of course, many SRI and SOX9 target genes are not yet known. The exact mechanism is not fully understood, and it's not yet elucidated, but we have some key points what we know already. So here we have one gene, and this is important. It's activated by the SOX9, and this is the AMH gene. AMH codes the anti-Mullerian hormone, AMH, and it causes the regression of the paramesonephric duct. Paramesonephric duct would give the female genital system. Of course, in males, it should be regressed. FGF9 is also a target of the SOX9. It is also important in case of the development of the genital parts in the male apparatus. We will see that the mesonephric tubules should penetrate the gonadal ridge, and this will be a very important step during the development. In case of the females, in case of the female cascade, we could mention the WNT4 gene, which is the ovary determining gene. This will indicate, or this will stimulate the transcription of the DEX1, and this DEX1 has an inhibitory effect on the SOX9. So SOX9 is a very important molecule in the cascade of the regulation of the male development, and in case of the female development, we should inhibit it, of course. So this is actually a very simplified version of the signal cascades of the molecular regulation of the reproductive development. And now we mention something what you have already heard about. This is the crossing over. So the crossing over happens during the first meiotic division. So this is what you have already learned from Embryology 1. And this was a very important event during the production of the gametes. And the crossing over means that the homologous chromosomes will form pairs, and they will become paired, and then they will exchange their genes. Very rarely, it could also happen then this gene which is exchanged is exactly that SRI gene or another very important gene causing testis development, or a gene which plays a very important role during the testis development. In this case, it could happen that this SRI will be translocated to an X chromosome, and if such a sperm cell will fertilize an X containing X cell, the sex chromosomes will be XX, but one of them will carry the SRI gene. And if the X chromosome carries the SRI gene, the testis development will start. In this case, we speak about XX males, and this is a very rare condition called the De La Chapelle syndrome. 90% of these XX males are SRI, sorry, that is a mistake, I should correct. So are SRI positive, so their X chromosome contains the SRI gene, but only in case of 10% they are SRI negative. In case of SRI negative cases, other genes are translocated, which play also a very important role during the development of the reproductive system of males, so testis development mainly. The other version is there when during that crossing over, this Y gene will be translocated away from the Y chromosome. Therefore, we will get a Y chromosome on which we don't have SRY gene. And if we don't have SRY gene, it doesn't matter that the sex chromosomes are X and Y. Without SRY, no testis development will start. In this case, the XY, the individual having XY chromosomes will become female. Of course, the genetically male, but phenotypically will become a female. For example, the Swyer syndrome, these malformations or disorders of the sexual development, we will see in case of the second lecture of this topic. And now we see already the development. First, we should focus on the source of the tissues. And this is the intermediate mesoderm. And this is what you have already heard previously when you've seen the differentiation of the mesoderm. And then we will do a very short recapitulation of Embryology 1, what we need for discussing the development of the reproductive system. Here on the left side, you see here the epiblast fate map. So this is the map from the stage of the bilaminar germ disk. So here we can see the level, the layer of the epiblast. And this map shows us what later would happen to the cells found here, to the epiblast cells found here. We need from this entire epiblast layer the intermediate mesoderm and the primordial germ cells, the PGCs. In this case, the intermediate mesoderm shows us a quite important area, giving rise not only to the reproductive system, but also to the urinary system. And this is what you have already heard about. This is the kidney development, and later we will also mention some points of the kidney development because this is closely related to the development of the reproductive system. If we continue, we will see what these epiblasts will happen to. And in this case, we will have, if I could change my, yeah. Here, in this case, you see also the epiblast here. And then here we see the process of the gastrulation. In case of the gastrulation, during the third week of the development, the epiblast cells will migrate through the primitive streak, as it is visible also here. And during this invagination, the epiblast cells will reach the layers below the epiblast, and from this middle layer, we will get the mesoderm. And during the differentiation of the mesoderm, we will get more parts of the mesoderm, like the paraxial mesoderm and the intermediate mesoderm or the lateral plate mesoderm. Now we concentrate on the intermediate mesoderm. Intermediate mesoderm gives rise to the reproductive system and the urinary system, to the urogenital system, with some exceptions. These exceptions we will mention a little bit later. So we need the intermediate mesoderm. On the next slide, you see what these structures will happen to. In this case, here we see the embryo, and here we see the yolk sac. After the gastrulation, the primordial germ cells, the PGCs, will be placed in the wall of the yolk sac. So after going through the primitive streak, the PGCs reach this region in the wall of the yolk sac, in the vicinity of the endodermal cells, and it is also important that it is the vicinity of the hindgut. These play one role in the story of the development of the reproductive system. The other role is of the genital ridge. The genital ridge is found, this brownish blackish structure here on the posterior body wall. This is the genital ridge. This is formed by the intermediate mesoderm on the medial side of the developing kidney here, and this is the genital ridge. The PGCs are now in this stage in the wall of the yolk sac, but during the beginning of the 5th week, they start their migration. Exactly, it is so that these PGCs, these primordial germ cells, will migrate with amoebic movement via the dorsal mesentery of the hindgut. So here you can see the hindgut. That is the mesentery attaching the hindgut to the posterior body wall, and along this mesentery, the primordial germ cells can reach the genital ridge, and then these primordial germ cells will populate the genital ridge. And then here we will get both the genital ridge, which is from the intermediate mesoderm, plus the primordial germ cells, which came via these dorsal mesentery. On the right side, you see the same structure, only in cross-section. And here we see also the dorsal mesentery. Along the dorsal mesentery, the primordial germ cells could populate the genital ridge, genital or gonadal ridge here. And on the lateral side, here we could observe the developing kidney. That is the second stage of the kidney development. On the next slide, we see also a summary about the structures, what we need for the development of the reproductive system. Here we see the developing gonad here. This light purple area is the developing kidney with the mesonephros, and the duct of it on its lateral side. It will be important for us. This is called the mesonephric duct or Wolffian duct, plays the key role in the male development, in the genital canals of the male development. On the lateral side of it, we have the paramesonephric duct or Mullerian duct, which has the main role during the female development. First, we will see the development of the gonads. In case of that events, what we've already seen, the primordial germ cells have already migrated along the dorsal mesentery. They have reached the genital ridge. In case of the genital ridge, on the surface we have epithelial cells. These epithelial cells on the surface will give the coelomic epithelium. The coelomic epithelium will proliferate and then will invade the mesenchyme found beneath it, and they will form the primitive sex cords. These events could be observed in both genders, both in males and in females. And until leaving the 6th week, so until the 7th week, no characteristics for the genders could be observed. Hence, we call this stage the indifferent gonad. In this case, we do not have any difference between male and female gonad yet. So this is the indifferent gonad in the 6th week. After the 6th week, changes will happen, and we will get the male development of male gonad, development of the testis, or development of the female gonad, development of the ovary. And these are the images what we will see later. So now we focus on the indifferent gonad. Then, in case of the male development, first we will discuss the male development. In case of the male development, here you can see the developing gonad, and on the surface, originally we've had this coelomic epithelium. The primitive sex cords, which were close to the surface, will get into the depth, so they will migrate deeper, and they will give the testis cords. On the 7th and on the 8th week, we can see them already. The testis cords found close to the hilus parts of the testis will become fine, and they will give a fine network of cell lines, and later they will give the rete testis. So this here is the rete testis, and now behind the rete testis here we can see the developing kidney again. This is one excretory tubule of the mesonephros. And here we can also observe these two ducts, which we have mentioned previously. So this is the mesonephric duct, and this is already the paramesonephric duct, and we will talk about them a little bit later. For the gonad, we do not need them. So for the gonad, we need the primitive sex cords, which will go into the medullary region of the testis, and they will become the testis cords. On the surface, they lose the connection with the surface because this connective tissue layer will be formed, and this will be the tunica albuginea. Later, in the 4th month, these testis cords will become horse-shaped. Inside these horse-shaped testis cords, we could already identify the Sertoli cells. These testis cords are solid testis cords without a lumen. The canalization of these testis cords happens only during puberty or short before puberty, and with that canalization, the seminiferous tubules will be formed, but this is only at the time point of the puberty and not before. In case of these testis cords, we can already identify the Sertoli cells. We have another characteristic cell type inside the testis. This is the Leydig cell. The Leydig cells will develop from the mesenchymal cells of the gonadal ridge. So the Sertoli cells derive from the surface epithelium, from the coelomic epithelium. The Leydig cells originate from the mesenchymal cells of the gonadal ridge. These Leydig cells show two different cell populations during life, and these are the fetal and the adult Leydig cells. Of course, in the developing fetal testis, we have the fetal Leydig cells. These fetal Leydig cells are able to produce testosterone already from the 8th week of the development, of the intrauterine development. These fetal Leydig cells do their function during the pregnancy and during the intrauterine development, of course, and the number of these cells will rapidly decrease short after birth. Then, after that, already in the postnatal testis, we will have the other cell population of the Leydig cells. They are the adult Leydig cells, and they will show again a very, very intensive increase in the puberty here. The Leydig cells produce the testosterone. testosterone is very important from point of view of the development of the male genital ducts. The testosterone could be converted to dihydrotestosterone by an enzyme called the alpha reductase enzyme, and this dihydrotestosterone will be also very important from point of view of the development of the external genital organs of the male. If we see further the male development, we should also mention the descent of the testis. So this is the process by which the testis will reach its position inside the scrotum. Originally, the testis starts its development in the retroperitoneal region behind the parietal peritoneum. So this blue layer is the parietal peritoneum, and behind the parietal peritoneum, in the retroperitoneal space, the testis starts its development in the lumbar, at the lumbar level, at the level of the lumbar vertebrae. Then the testis should descend, of course, until the 12th week. It doesn't happen anything, or already in the on the 12th week, the testis is in the inguinal region before it should be above. So here in the lumbar region. So we are already in the 3rd month, and testis is found here. Independently from the descent of the testis, one process of the peritoneal cavity is visible. So this is protrusion of the peritoneal cavity. This is the vaginal process. It is not dependent on the descent of the testis, so, but they happen at the same time, or they are visible at the same time. It does not happen anything until we reach the 7th month. If the embryo is 28 week old, the testis starts its here already on the right side. It starts its descent through the layers of the abdominal wall, and with the descent of the testis through the abdominal wall, the inguinal canal will be formed. So this is also the formation of the inguinal canal as well. So there's no inguinal canal before, but testis develops the inguinal canal with its descent. The testis passes through the layers of the abdominal wall, and it pushes every layer of the abdominal wall in front of it. So in case of the descent of the testis, we have a very important structure which should be mentioned. This is the gubernaculum testis. The gubernaculum testis, I can show it on a previous picture as well. So the gubernaculum testis is between the caudal pole of the testis and the inguinal region in this stage. The end of the gubernaculum testis is attached to the developing abdominal muscles, to the developing between the developing external and internal oblique abdominal muscles. We find this lower part, and then it starts its descent. In case of the 12th week, it's already close to the entrance, and of course during the 7th month it starts its descent through the layers of the abdominal wall. And here we also see this gubernaculum, which will actually direct the descent of the testis through the through the anterior body wall and the later inguinal canal. In case of the end stage, of course, if the embryo is 33 week old, the testis has already arrived in the scrotum. The vaginal process changes. loses its connection to the peritoneal cavity, and only this will remain. This is the tunica vaginalis, and this will be found on the anterior side of the testis and will be composed of, or is composed of, two layers. We have a visceral layer found on the anterior side of the testis, and a little bit far away we have the parietal layer. So this is the descent of the testis. It is controlled by androgens and AMH, so it is also under the influence of hormones. If we see that descent, we could understand which layer of the capsules around the testis could be ordered to the layers of the abdominal wall. So here we can see the layers of the abdominal wall, the external oblique, internal oblique, transverse oblique, and its fascia. And these layers will be pushed by the testis during its descent, and therefore these layers could be seen, or the derivatives of these layers could be seen around the testis. They are the capsules around it. Of course, the vaginal process normally loses its connection with the peritoneal cavity, and only this tunica vaginalis remains. But it is also possible that something went wrong, and we will have some pathologies in this case. First, we have this cryptorchidism. Cryptorchidism means that the testis does not reach the scrotum until the week 33, and in this case the testis could be either inside the abdominal cavity, or inside the inguinal canal, or inside the spermatic cord, or at the level of the spermatic cord. After birth, generally it will be solved automatically, so there's actually only postnatal descent of the testis, but in some cases, in 1% of male births, the surgical correction should be done. The other possible pathology is the inguinal herniation. In this case, the vaginal process does not lose the connection, or now tunica vaginalis does not lose the connection with the peritoneal cavity. Therefore, it remains here, and then we have the possibility for getting herniation here. So this is the congenital inguinal hernia. We have another possible version where, when this connection only partially remains, it means that we will have an irregular connection between the tunica vaginalis and the peritoneal cavity, and in this case cysts can be on the way between the peritoneal cavity and the tunica vaginalis, and these cysts could secrete fluid, and hydrocele could be visible in this case. Of course, it could be even larger. This was the development of the testis, and now we see the development of the ovary. Of course, this is also after the indifferent stage, so after the 6th week, and here we see what here happens. So here we are in the on the 7th week, and in this case we see also the structures originating from the primitive sex cords. Primitive sex cords. They are the medullary cords, which in case of the testis will remain and will give the seminiferous tubules later. But in case of the ovary, these medullary cords originating from the primitive sex cords will disappear, they will degenerate. And here in this area, we will see blood vessels, nerves. These blood vessels, nerves replace the site of these degenerating medullary cords. And then this area will be the medulla of the ovary. In a later step, from the surface, the proliferating coelomic epithelium will give rise to secondary or cortical cords. These are secondary or cortical cords, characteristic only for the ovary. Such secondary or cortical cords that do not exist in case of the male gonad, in case of the testis, only here. This here does not lose the connection with the surface, and it will be also on the rim or close to the rim of the ovary. So here we see them. These cortical cords during the 3rd month, what we could not see on these pictures, in the 3rd month, they will split into cell groups, and they will surround the developing germ cells. Hence, they will give the primordial follicles, and then they develop further, of course. And here in this stage, which is already in the 5th month, this contains already primary oocyte with the follicular epithelial cells around. So here we do not have any cords in the medulla, but only the blood vessels and the nerves. Descent of ovary, which is not so pronounced as that of the testis was. We will talk about it later, but it's a very, very quite small descent in this case. If we turn to the development of the genital ducts, in this case, we have also an indifferent stage. In case of this indifferent stage, we will see 2 different ducts. We have the mesonephric duct. This is what you already know. The mesonephric duct is related to the kidney development, and that is the structure related to the 2nd stage of the kidney development, to the mesonephros. And on the lateral side of the mesonephros, we have a structure having a lumen that is called the mesonephric duct. This will play the main role during the male development. On the anterolateral side of it, we have this brownish orange colored structure. This is the paramesonephric duct or Mullerian duct. This is formed by the craniocaudal invagination of the coelomic epithelium. It means that from the surface, simply first we will get a groove, and then the groove will be closed. And this is craniocaudal because it starts here at the upper pole of the mesonephros, and it will go down in caudal direction. And then at the end, both ducts will open into the urogenital sinus. And this is what we will discuss later. So the end is always in the urogenital sinus. The cranial portion of the Mullerian duct opens freely in the coelomic cavity. Here we can see it. And then if we go from this indifferent stage in direction of the already defined stages, first male development, we will see. Here we see what to these ducts will happen. In case of the male development, the mesonephric duct will develop further and will differentiate into different parts of the male genital system, and the paramesonephric duct will undergo an atrophy. These events are controlled by hormones. the testosterone acts on the development and differentiation of the mesonephric duct, testosterone by the Leydig cells, anti-Mullerian hormone by the Sertoli cells is responsible for the atrophy of the paramesonephric duct. Okay, in this case, is it so? If we see the details from the mesonephric duct here, this is the mesonephric duct, the developing, differentiating mesonephric duct. Here we have already the testis cords plus the reta testis, which we have already seen in case of the development of the gonad. First, we need a connection between the mesonephric duct and the reta testis. This is made by tubules of the mesonephros. They are mesonephric tubules. At this level, we find the epigenital tubules, and the epigenital tubules will be the efferent ductules of the testis. The later stage you see here on the right side. So here from these epigenital tubules, we get the efferent ductules of the testis here. The tubules found above the epigenital tubules will degenerate and will disappear. The tubules found below the epigenital tubules, called the paragenital, will give an embryological remnant, which is here visible. This is the paradidymis. So paragenital tubules will become paradidymis. Then here we have this portion which receives the efferent ductules here. It will become very coiled, and this will be the epididymis, exactly the ductus epididymidis here, and it will be continued here and here in the vas deferens here. And then the vas deferens in this stage opens here already, opens here into the urogenital sinus. Near the junction between the derivatives of the urogenital sinus and the vas and the mesonephric duct, one protrusion will occur on the 10th week of the development, and this protrusion will give the seminal vesicle. And then this protrusion will define also its duct, the excretory, and the mesonephric duct beyond the origin of the seminal vesicle will be the ejaculatory duct. On this picture or on this slide, we can see also remnants of the Mullerian duct. So in case of males, the Mullerian duct regresses. Only the prostatic utricle, the utriculus prostaticus, and the appendix testis will remain, and they are only embryological remnant. We would have 2 more slides, but with this slide, we will start the second lecture of the development of the reproductive system. Thank you for your attention.